Hi, this is Mr. McLean. In this video, I'm going to take you through the first part of Chapter 7, where we're going to take a look at what the membrane structure actually has, and we're going to take a look at some of the different functions of the various parts. I'm going to shut my webcam off here so that we can focus more on some of the things I want to try to draw. So let me see if I can get rid of me. And I'm gone. All right, so one of the first concepts we really want to make sure that we understand is chapter 7. We start to see how the molecules are put together to make up the membrane. And when we use this term amphiotic, what that's talking about is the fact that our phospholipids, which are the main molecules of the membrane, are going to have opposing characteristics. So at one side you're going to have hydrophobic, so that is obviously going to be the non-polar end, and then you're going to have the hydrophilic, which is going to be polar. So in a slide coming up here in a second, I'm going to show you how those different characteristics actually exist. So if we look at this image right here, this is our bilayer. Now obviously there are some other things that are going to be in there that we'll see, but this is the majority of what makes up the bilayer. And if you take a look at what our individual phospholipids actually look like, what you're going to notice is that we have the tail regions which are hydrophobic. And based on this image here, you're going to see that there is no water really found through here. So this area right through the middle, all through these tail regions, is very hydrophobic. So it's difficult for things that are polar, such as water. What we do notice is the head regions are polar. So that's why we see water on the inside and the outside of the membrane. That's going to be focused on that. Now, if we take a look at it a little bit more closely, one of the other things that we're going to notice is that we have a lot of different proteins that are embedded in that particular membrane. So in this particular area, one of the things that we will see is that those proteins serve a variety of different functions. Now, as those proteins are embedded in that overall composition of phospholipids and cholesterols and sugars and things of that sort, one of the things that this slide is focusing on is that each of these individual phospholipids can kind of like move and shuffle past each other. So the analogy that your book talks about is being in a very tight crowd and just kind of like, you know, pushing past somebody to get to this way, and they sort of swap positions with you. So you kind of have this back and forth kind of pushing motion. And that is what we're noticing. That's the lateral movement. So there's a lot of lateral movement. There's a lot less flip-flopping movement, where they're going to go from the extracellular side to the cytoplasmic side. Another really, really important thing on this particular slide is talking about the phospholipid composition. So if you look at this image here on the left, you're going to notice that these are going to be unsaturated. And when you have unsaturated, obviously that should immediately tell you that you're going to have the double bonds. And when you get those double bonds in that carbon chain, what that's going to end up doing is it's going to give you these different bends. So unsaturated is missing hydrogen, and as a result of that, these bends are created. And what that does is that prevents this structure from packing together really, really tight. So by having a little bit of that space in there, it keeps fluidity. So an organism who has this type of composition is most likely going to be able to tolerate a cold environment. Whereas if we slide over to the right, we find an organism where we have a much greater percentage of saturated fatty acids making this up. And that's what the straight chains are going to show you. So they have a greater viscosity, or they're viscous, which means they're going to resist flowing. So as a result of that, they pack together a little bit tighter. So this one is probably more consistent with an organism that lives in a very warm environment. So I would say this is the hot environment setup. This would be the cold environment setup. Pretty important for the test. Another factor that also is influenced by the temperature is the composition of these steroids inside of here. And if you recall, the general composition of a steroid is going to have the 4 carbon rings. And this specific steroid is cholesterol. So the cholesterol is serving 2 functions. Function number 1, in this setup here, it's a lot of saturated fats. So the cholesterol is kind of keeping a little bit more spacing in that, if you would. So it doesn't become overly packed together and really, really tight. Whereas in other applications, you're going to find out the cholesterol is actually going to kind of hold it together. So if you had a setup that was more similar to this, the cholesterol would kind of hold things together for us. So it serves a dual function in that. This slide's just talking about the fact that not only do our phospholipids move, but our proteins also move. So we can see evidence of that. And the movement of those proteins is going to show us that what they're ultimately being controlled by is the way in which they're being distributed on the perimeter of the cell, is how they're processed and how they're finalized as they go through the process of dealing with the Golgi apparatus. So our membrane, this picture right here is a very, very important one. I would reference this for the test if I were you. The structure is showing us that internally we've got this network, which we learned about, that microfilaments right below that. And what they're doing is they're kind of giving resistance so that this doesn't get shredded, because otherwise it would want to pull apart. And then we can see a variety of different types of proteins. If they're on the edge and not really penetrating all the way into the interior portion, we refer to those as peripheral proteins. If it's something that goes into the lipid bilayer, we refer to it as an integral protein. Now, an integral protein can kind of do this, where it only goes in maybe a portion of the way, or it could go all the way across. So if it passes all the way across, then we refer to that more specifically as a transmembrane protein, which is integral. Zooming in over here, we have carbohydrates that are attached to the lipids. So in this case right here, you can see the sugars hooked onto that. So that makes up a glycolipid. Over here, we're going to see proteins with sugars hooked onto it. That makes up a glycoprotein. Those sugars on the surface serve a major function as far as being an indicator of cell cell type and communication. So when we start looking at how cells are going to be recognized, a lot of times we rely on the external carbohydrate setup. Okay, so other things, you got your ECM on the outside. The ECM has connections, so like we have our fibronectin, which is going to anchor that ECM to the internal proteins. And like I said, you have a lot of proteoglycans in the setup. So that's the general structure. If we look at a transmembrane protein, one of the really critical functions that they can serve is they can serve as a passageway for certain types of polar molecules or ions. And the way that that's created is that it creates kind of a hydrophilic interior inside of those helixes that are established inside there. So if we look at what the proteins are actually doing, we can focus on 6 big things. And we have them listed there, and we're going to run through the pictures pretty quickly because I think they make it a little bit easier. Okay, the first one, I'm going to kind of hold off on a major explanation of this because this is really the big topic of the chapter, but that is they serve as a point of transport. So really, this picture is showing us 2 different scenarios. Scenario number 1, we have molecules going down a gradient. So maybe these are ions or something that's polar, and this would be passive. Over here, energy is being invested, and we're taking something from a low concentration and we're making an even greater concentration. So this is active. Both rely on proteins. All right. Another function of the proteins is that they play a role in chemical reactions by serving as the enzymes. So embedded in the membrane, you have these active sites, and the active sites will take in the substrates and they will catalyze the formation of product. Okay, so those are our reactants being formed into products. And we'll see in chapter 8, we spend a lot more time on the functions of enzymes. Chapter 11 in this book talks a lot about cellular communication. So in this case, our signaling molecule, which is a ligand, that's a general term for any signaling molecule. So it might be something that's part of the nervous system. It might be a hormone. Any molecule that is serving as a signaling molecule is a ligand. And this protein has an active receptor site right here. So once this pairs up and it fits, kind of like a lock and key type of fit, it will initiate what's referred to as a signal transduction pathway. We'll learn much more about that in a future chapter, but the idea is that we're able to communicate signals from the outside to the inside. In this particular passageway, this molecule is probably not able to make it across. So what it's going to do is it's going to rely on the fact that maybe it's polar or maybe it's just too big, but it can set off kind of a chain of events. I like to think of it almost like the domino effect. Once you set off the first event, the next couple will follow, and ultimately you'll get your result. All right, cell recognition, I already mentioned that a little bit there, but these are those sugars. So when you start looking at the immune system, for example, and how our body is able to recognize self versus non-self, and then the issues that go along with that. Glycoproteins serve as a cellular, I guess you could say, tag on the surface that lets you know what cells are yours, basically. We also have cellular junctions that form between different proteins. So when you're thinking about the formation of a tissue. And then the last one on here, we had mentioned during our tour of the cell, but if you look at it, okay, so right here, this is my internal cytoskeleton, and here is a transmembrane protein setup, and over here we have fibronectin, and then this thing, it's probably collagen. So the idea is our ECM and our internal cytoskeleton and the membrane as a whole are able to work as a unit because of the combinations between those different types of proteins. So each of those things are really critical. Just to kind of summarize a few final points in here, this statement right here, talking about the recognition, the carbohydrates, it says even members of the same species are going to have different setups. So unless you have an identical twin, your carbohydrate characteristics on the outside of your cell in many ways are going to be very unique to you. And each of those faces have distinct characteristics, as I was kind of getting to earlier, and that's determined by how those proteins are assembled, both at the ER and at the Golgi, because as we recall, for this type of setup, these proteins were most likely manufactured on the rough ER. And when they were made on a rough ER, they were then passed through the endoplasmic reticulum, and then they would be packaged with portions of some of that membrane, making up a vesicle, and then they would have made their way to the cis face of our Golgi, and they would be modified, and eventually they would pass out of the trans face, and as a vesicle, they could eventually either be secreted or become part of what we were talking about. So there's an example of a transmembrane glycoprotein that is appearing inside of our plasma membrane, or a glycolipid, which is happening there as well. All right, so hopefully that clears up a couple things about the structure. I'll put out a few more videos in regards to some of the passive and active movement. Thanks.